Good morning, students. Today we are going to start the next section. That is membrane pumps, carriers, and channels. Last class we have learned till about the functions of plasma membrane, right? So today we are going to see different types of carriers, pumps, and channels. Three different types of membrane pores are present in the plasma membrane to control the permeability. Which are those pores? They include pumps, carriers, and channels. First, we are going to learn about pumps. What are these pumps? They are certain enzymes which can pump ions and solutes across the membrane. How? Pump itself means that it needs energy. So, by utilizing energy from ATP, that F is missed there. Energy from ATP or light or some from other sources, it pumps ions and solutes across the membrane. What this pumps? Okay, what's the significance of this pump? Only because of this pump, the itself can maintain the concentration gradient. For that, to understand that this is a plasma membrane, and what is the charge outside? Outside, and this is the inside. What is the charge outside? Outside is always positive. Negative, sorry, inside is always negative. This is the pattern of the charge outside and inside of a membrane, cell membrane. And this distribution, this condition is maintained mainly with the help of the pumps. And we know that potassium is the main important ion that present inside the cell, and sodium is the ion that present predominant ion that present outside the cell. And they play an important role in maintaining the membrane potential. Regarding that all, I will explain later. Now you understand only this much. For maintaining the concentration gradient across the membrane, what plays significant role? The pumps play significant role. Example for such a type of pump is F0, F1 ATPase. Its picture is shown here. It is present in the inner membrane of mitochondria. It is present in the inner membrane of mitochondria. It is involved in the synthesis of ATP, ATPase. So that's why I called it ATPase enzyme. F0, F1 ATPase is an example for pump. Another example is sodium potassium pump. It is present in nerve and muscle cells because these nerve cells and muscle cells are the most excitable cells. So there, the sodium and potassium pump is very significant. Another example. Next, we are moving to carriers. Carriers, I will explain to you. Carriers means what? Carriers means what? They are integral membrane proteins. It's completely spanning the lipid bilayer. And they provide some passive pathway. Passive means what? Passive means it doesn't need any sort of energy for transporting that molecules. And its transport occurs in the direction of down the concentration. Down the concentration means from where to where? From high concentration region to less concentration region. That is the direction of the transport by carriers. Example for such carrier is membrane-bound carrier proteins involved in facilitated diffusion. In facilitated diffusion, certain proteins are involved. They are examples for carriers. Next is about channels. As the name itself make it clear that channels means something that is having a central pathway. Okay. Same thing also here. Channels. Channels, they are again integral. Integral means it's spanning completely. Integral proteins and through which ions can diffuse passively without the requirement of any energy. These channels are ion specific. For the particular ions, there will be particular channels itself. And it close transiently. It can open and close depend upon certain stimulus. And these channels are formed by, sometimes it is formed by a single protein, or sometimes it is formed by the aggregate of many different proteins having a central channel through it. Then they are called gated channel. Why it's called gated channel? Because it can open and close. That's why they are called the gated channel. Examples are sodium gated channel is there, potassium gated channels are there, calcium ion gated channels are there, and they are all usually found in nerves and muscles. Then we are going to see different types of gated channel. Gated channels are of three types: voltage gated channel, chemical gated channel, and stretch activated channel. Voltage gated channel means when it open. Suppose voltage gated channel means there is a particular voltage is existing in the membrane. If whenever a change happens to that voltage, then that particular gate opens. Okay, that is about voltage gated channel. Then chemical gated channel means what? Means that gate opens when the chemical gated channel opens whenever certain chemicals bind to that. They are called the ligands. Ligands are simply certain ions. Okay. So whenever such ions bind this gate, that time it opens. Such type of channels are called chemical gated channel. That's what is stretch activated. Stretch activator means they are such type of some type of channels. They open when it is stretched. When its membrane gets stretched, then that time it opens. Example is stomata. Stomata is example for that stretch activator. We know that stomata controls, regulate the transpiration. This is the expel or to release excess amount of water present in the leaves, right? We know that, and I don't need to explain about the stomata. Stomata is example for stretch activated channel. Next, I am going to show you one video in that you can see almost all type of channels.
Layers of phospholipids. The hydrophobic nature of the cell
Okay, this
membrane
is
makes it
plasma
intrinsically
membrane.
permeable to small
This is simply plasma membrane. First, in the beginning part of this chapter, I have told that small molecules, water, can easily pass through the plasma membrane. The size of the particles increases, then it cannot cross the plasma membrane. Okay, it's showing
nonpolar
here.
and uncharged polar molecules, but
They
non-permeable to
can easily
large polar
pass.
molecules and charged particles. Charged particles
But look, the charged large molecules, they cannot cross this plasma membrane. For these one, we need the different types of channels, carriers, and pumps.
such as ions must use special channels to move through the membrane.
Here we can see the sodium channel. It is specific for the sodium ion.
Transport of a molecule can be passive or active.
Passive transport and active transport is showing here, and this is a simple picture to show that this is a slope, so it doesn't need any energy to roll this ball down on the slope, but
Does not require energy input because it moves the molecules downhill. For example, from higher to lower concentration.
look here, similar way this is a picture show that here so much of concentrated, if we consider concentration, here the concentration is high and here the concentration is very low, so particles move from region of higher concentration to lower concentration. So this is passive, doesn't need any type of energy.
Active transport, on the other hand, moves
This is the active representation. Here It is a means it's not a slope, it's against, we need to move this ball against this slope, so it needs energy. That accounts for the active transport. In this picture also we can see that this is the less concentrated region, is the high concentrated region. And if a molecule from this low concentrated region need to move to the higher concentrated region, then it requires energy. Okay.
The molecules against their gradients and therefore requires energy expenditure. Ion channels permit passive transport of ions.
This picture showing two channels, sodium channel and potassium channel.
These are transmembrane proteins that form pores for ions to pass through. Most ion
Showing passive transport.
channels are specific for a certain type of ion. Ion channels can be classified by how they change their open, closed state in response to different factors of the environment. Common types of ion channels include leak channels. These channels are almost always open, allowing more or less steady flow of ions. Examples are potassium and sodium leak channels in neurons. Ligand-gated ion channels. These channels
Ligand gated, I already explained to you. Certain substances are coming and bind. Upon binding, then that gate is open.
open upon binding of a ligand. They are most commonly found at synapses, where neurons communicate via chemical messages, or neurotransmitters. An example is the GABA receptor, a chloride channel located on postsynaptic neurons. It opens upon binding to GABA, a neurotransmitter released by the presynaptic neuron, and allows chloride ions to flow into the cell. Voltage-gated ion channels.
Voltage gated. Voltage gated also I explained to you, when there is some change happen to the voltage, usual voltage, then that gate will open.
Channels. These channels are regulated by membrane voltage. They open at some values of the membrane potential and close at others. These are the channels that underlie action potentials in neurons and cardiac muscles. Active transport of ions is carried out by ion transport.
Next is pumps for active transport. That is means they pump, they let the ions to move against the concentration gradient. We know that sodium is the predominant ion outside, potassium is the predominant ion inside. So here somehow means we know that from a region of higher concentration to low concentration, everything can move. So sodium also can move from this region to this region, because here the concentration of sodium is very less. Okay. And it disrupts the innate potential voltage across the plasma membrane. So we need to expel, or we need to remove the additionally entered sodium to the inside of the cell. For that we need the help of pumps, and that pumps, as it is working against the concentration gradient, it needs energy.
Transporters or ion pumps. These are transmembrane proteins that pump ions against their concentration gradient using cellular energy, such as ATP. Most notable example is the sodium-potassium pump, which maintains the resting potential in neurons by pumping 2 potassium in and 3 sodium out of the cell. Another type of ion transporters, known as secondary transporters, do not use.
Okay, only up to here we want for our topic. Okay, then I think channels, pumps, and then which one? Channels are there, pumps are there, and carriers are there. All are clear to you. Okay. Next is about transmembrane transport. Transmembrane transport. What is transmembrane transport? It involves the exchange of material across the cell membrane, traversing the lipid bilayer. Yeah, just before, in the former slide, we have seen about the different types of carriers, then pumps, then channels. All these are certain proteins that is helping the transport of materials across the membrane. So which are the, now we are going to see which are the different process. that is helping for the transport of materials across the plasma membrane. That include diffusion, osmosis, facilitated diffusion, active transport, then bulk transport. Which one we are going to learn now? First one is diffusion. Diffusion is very simple. I think almost all of you know about what is diffusion. Still then I am explaining it. Diffusion. What is diffusion? It is a movement of substances from the region of higher concentration to a region of lower concentration. Here you can see a picture in this side. It is having more concentration. This side having less concentration. After some time, as we notice, we can see that the both sides have reached equilibrium with regard to the concentration. They attain that equilibrium because of diffusion. How it what is the pattern of movement? The substances move from higher concentration to the lower concentration. By that way, they attain that equilibrium. This is one method. Plasma membrane. It is impermeable to charged ions, but it moves across the plasma membrane. How? Even if the plasma membrane is impermeable to charged ions, it is moving. How? It is through ion channels by the process of diffusion. Through ion channels by the process of diffusion. Certain ion channels will be there, and through that channels ions move according to their concentration gradient. Okay. Next process is osmosis. Osmosis and diffusion are two related processes. What is the difference of osmosis when compared with the diffusion? For osmosis, here adding a semi-permeable membrane in between two separated compartment of solution. Look, what is the peculiarity of solution on both sides? Here we can see more solvent and solute particles are very less. And in this side, what about the solutes are more and the solvent is less. So, in osmosis, what is happening? In which place the solvent's concentration is more, from that region solvent only is moving. Solute cannot pass. Why? Because there is a semi-permeable membrane. That solute cannot cross this semi-permeable membrane. And what only moves? That solvent, that liquid part only moves to the semi-permeable membrane to the other side. So osmosis, it involves only the movement of water. One more term we need to understand, that is water potential. What is water potential? Water potential means it's the tendency of the water molecules to move from one place to another depending upon the concentration gradient. Where water is more, from that place to the other place where the concentration of water is less. To that place it flows. That is water potential. So, as a definition for osmosis, we can tell like that. Osmosis means it is a movement of water molecules from a region of higher water potential, means higher water concentration, from higher water potential to a region of lower water potential, means lower water concentration through a partially permeable membrane. Very clear. It's also very simple. Next is facilitated. Facilitated. That term itself indicates that some facilitator will be there. It is otherwise called carrier mediated diffusion. There will be certain additional molecules be there to help the diffusion process. Diffusion, we know that it is a passive. It doesn't need any type of energy requirement. It only happened in the direction of concentration gradient. So which molecules relay this type of transport? Usually sugars and amino acids relay this method, facilitated diffusion. Yeah, that you have to memorize. Then what are the features of facilitated diffusion? It is substrate specific. Substrate means that carrier molecule or the carrier that present in, or helping that diffusion is highly specific. So see for which type of substrate it can transport. And the next feature is that it is competed by structurally similar substrate. If any other substance that is similar with the shape of the substrate is present in the medium, it will compete with the carrier for binding in its binding site. In that way, it can inhibit the actual substrate from binding that carrier protein. This is another feature. And next feature is that its function, that carrier protein's function is affected by pH. The variation in pH greatly affect the performance of that carrier protein. The next feature is that the transport that is performed or done by the carrier protein is taking place in the direction of concentration gradient. And the last feature is that the activity of that or the function of that carrier can be inhibited by enzyme inhibitors like sulfhydrate blocking agents. These are the features of this facilitated diffusion. Okay. Next, active transport. Active, as the name implies that active means it requires some energy. What's the difference of this active transport from other two types, other different types of transport from diffusion, facilitated diffusion? Because it is taking place against the concentration gradient. Here, the transport is taking place against the concentration gradient. As it is taking place against the concentration gradient, it requires energy. So, usually it is from ATP. Next, and this active transport is influenced or affected by certain conditions. First one is when the cell is too much cooled. That means if it at temperature 2 to 4 degrees Celsius, then the active transport will not takes place effectively. And also if the cell is subject to poisons like cyanide, iodoacetic acid, then that time also that active transport is very much hindered. And also when there is no enough amount of energy, we know that active transport require energy for transporting molecules across the plasma membrane. So whenever there is no sufficient energy, that time also active transport will not take place properly. Example for active transport is sodium potassium transport, and that takes place with the help of a protein, a pump called sodium potassium pump. Next we are going to learn about different types of carrier proteins. First one is uniport. Uni means single transport, single molecules transport. In that way we need to understand. Look in this picture and then we can understand uniport. Uniport means it is transporting a single solute from one side to other side in a single direction. That also takes place from a region of higher concentration to a region of lower concentration. From that transport itself is taking place from higher concentration concentrated region to lower concentrated region. Next, co-transport or couple transport. Couple means it itself means that two molecules are transporting across the plasma membrane. Such type of carrier protein. What is co-transport? I already told you. Simultaneously, two molecules are transported here. In this picture, we can see that two molecules are transporting. This co-transport is of two types, symbiote and antiport. Symbiote, what is symbiote? Symbiote means it binds two dissimilar molecules and transport in the same direction. That is important. Same direction. That is important. Example is sodium glucose transport. Both are transporting in the same direction. And what is the specialty of symbiote? Specialty is that it can move solutes even against the concentration gradient. How it happens? Do you understand that? I will show you. This symbiote, even if it doesn't need any energy, and it always takes place downhill in the direction of concentration gradient, still then it can transport solutes against the concentration gradient. How? That I am going to show you. If this is the plasma membrane, and the concentration is this much for this molecule, and here it is only this one, and we need to transport it against. How it happens? Anyway, we understand that it requires some energy. From where that energy is obtained? That energy is obtaining from the downhill transport. Uphill transport of some other molecule that is taking place in the direction of concentration gradient. Clear? Because here only the transport is against the concentration gradient. Means this transport. This is only taking place against the concentration gradient. For that, energy is required. From where the energy required for that process is obtained, that I am explaining. It is coupled with the transport of some other molecule, and that molecule's concentration is more here. So, and its concentration is less in this side. So, its transport occurs in the direction of concentration gradient. So, it is a downhill transport. It doesn't require energy, but it can release energy. And using that energy, the transport of this molecule against the concentration gradient is taking place. That's what I explained in this sentence. Symbiote can move solutes against the concentration gradient coupled with the downhill transport of other solute. Clear? Next is antipode. Anti means opposite. Here also two molecules are transporting, and those two molecules are transporting in opposite direction. Example, sodium calcium co-transport. Okay. Both the molecules are transporting in opposite direction. Next, I am going to explain about the sodium potassium exchange pump that is doing active transport, and that enzyme or that protein is called sodium potassium ATPase. What is the significance of this pump? Because of this pump, the membrane maintains its potential. I already explained to you that the plasma membranes outside is positive and inside is negative, and the resting membrane potential is almost minus 70 millivolt. For maintaining this pump is playing the significant role. For the function of this pump, ATP is required. So we are going to see how it functions. um, this is the plasma membrane, and this is outer side, and this is the inner side. The outer side, which is the prominent ion, it is the sodium. These red squares are sodium, and which is the prominent ion present in cells inside, it is potassium. And as sodium is more in outside, Then what is the general tendency? Sodium has a general tendency to diffuse towards cells inside in the direction of concentration gradient. Because of this diffusion, there is chance that some sodium have entered the cells inside. And also similar to that, potassium, what it has the tendency, it has a tendency to migrate or diffuse to cells outside in the direction of its concentration gradient. Okay. So by that way, there is chance to have some potassium outside the cell. If it goes on continuously for long, it may disrupt the normal membrane potential. We need to prevent that. For preventing that, there's function this sodium potassium ATPase and how it is functioning. And all the proteins, it have several conformation. For understanding that, first I am going to tell you that protein have different level of organization. Primary level, primary, that is primary structure, it is the extended structure. Then tertiary structure is there. Tertiary structure means it is the folded pattern of the protein. Then secondary, then tertiary is there. Tertiary means this folded proteins again folded to give a complex structure. There is a tertiary structure or 3D structure. And then quaternary structure also there. Quaternary means one or more tertiary structure get aggregated to give a single protein, to form a single protein, that is the quaternary. So different conformations are there. So we are starting with here with this conformation. In the present conformation, what is possible? Possible for 3 sodium to come and bind in different binding site of this enzyme. As 3 sodium binds in 3 different sites, this enzyme get phosphorylated by ATP and ADP is released. This is phosphate. This one is phosphate. Okay. And again the conformation change, and because of that change, what happened? The phosphate that is attached with the enzyme leave the enzyme. And as it leave, what happens? The potassium that has diffused out to the cells outside, among them 2 come and bind this enzyme. Only 2 is coming. And 3 sodium that have previously bound to the enzyme left the enzyme to the cells outside. Okay. So while releasing 3 sodium is taking only 2 potassium. That is important. You have to attend that, because while releasing 3 sodium, it is taking only 2 potassium. Again conformation change, that folding pattern again change, and then same what it do next? The 2 potassium is released to cells inside. This is one cycle. Again the cycle repeat. Again the cycle repeat means what next to happen in this conformation? Again 3 molecule of sodium come and bind this site. After this sodium bind, then what next to happen? It need to be phosphorylated by ATP. After that conformation change, it changed to another conformation. Potassium is binding. Then the phosphate group attached with the enzyme is leaving that enzyme, and conformation again change, and that potassium is releasing to cells inside, In that way, that cycle is repeating and repeating through many cycles until the membrane comes back to its normal resting membrane potential. Again, I am this is the functioning or working of the ATP, sodium potassium ATPase pump. And along with that, I want to add one important point, because while explaining the process itself, I point you that this enzyme is releasing three sodium ions to outside. The same time, it really take only two potassium from outside to inside. That means it is taking more positive charge to outside for maintaining the outside positive, and it's taking only less number of positive to inside to keep the inner side of the plasma membrane negative. Clear? In that way, it is helping to maintain the potential of the plasma membrane. Inner side should be negative, outer side should be positive. For keeping that potential, for maintaining that potential, it is releasing 3 sodium to outside, and in return only taking 2 potassium to inside. Okay. That is the relevance of that exchange. Next is coupled transport or co-transport of metabolites. Means two molecules are transporting at the same time in the same direction. Co-transport usually occurs for amino acids, sugars, and other metabolites. And usually its transport is coupled with the sodium ions, along with the sodium ions. What's the positive factor, favorable factor? Favorable factor is the steep concentration gradient of the sodium built up by sodium potassium pump. We just only have learned about the sodium potassium pump, and as we learned that, we understand that the pump is meant for maintaining the steep concentration gradient of sodium and potassium across the plasma membrane. And in the outer side of the plasma membrane, the concentration of sodium is more. So it has the general tendency to diffuse to inside. By taking account of that property, some other molecules also the cell or that a particular enzyme can transport from outside to the cells inside. That is the positive factor, depending on the steep concentration gradient built up for sodium or potassium. Using that, some other molecules also transporting to cells inside. Next, we are going to see that process. This is the carrier protein. Sodium, we know its concentration is more in cells outer side. What is happening because of that? Glucose also present. Whenever glucose is present, what happened? One more thing we have to understand is that there's a steep concentration gradient is there in this direction. Means concentration for sodium is more in this side, and this side concentration of sodium is less. So what happens next? The glucose first come and bind the enzyme or the carrier protein. As glucose binds, following with that one sodium also come and bind another site of this carrier protein. And these binding change the conformation of this enzyme or that carrier protein. And it fades to inside. As it fades inside, can release the glucose and sodium. In that way, transport glucose from cells outside to cells inside by taking grant of the constant concentration gradient of sodium. That concentration gradient is actually developed by or built up by sodium potassium pump. Very simple. Only this much. Again it returned back to its original conformation after releasing the glucose and sodium. Once more I will repeat. Whenever glucose is present in the medium, that sodium binds to the carrier protein and the glucose also bind to carrier protein. Its conformation change, it phase to its that binding site phase to cell's inner side, release glucose and sodium, and after releasing that glucose and sodium, again conformation change and it again phase to cells outside. That is co-transport of glucose coupled with the sodium. Okay. Next, we are going to understand about bulk transport. Bulk, that term itself indicates that it is a large scale transport. Large scale transport into or out of the cell through the plasma membrane. It is of two types, endocytosis and exocytosis. Endo means to the cells inside and exo means from cells inside to cells outside. First, we are going to learn about endocytosis. Means something is taking to cells inside from outside. The cell and other substances. And for that, first plasma membrane invaginate to form a pit. And then that pit size increases. And then it detach to form a vesicle and take in completely it take in the substance that is present outside. This is endocytosis. By forming the vesicle, it entrap the material that present outside, and that vesicle is formed from the plasma membrane itself. That is endocytosis. Simply, what is endocytosis? Endocytosis is the taking in of substances from cells outside to inside by the formation of vesicles. Okay. Types of endocytosis. Endocytosis are of mainly three types: pinocytosis, phagocytosis, and receptor mediated endocytosis. Each one we will learn in the next slides. Exocytosis also I am explaining here. Exocytosis means the substance from cells inside is transporting or releasing to cells outside. This is the material or substance that need to release outside. This is releasing in this way. First, it is trapped in a vesicle in cells inside, and then this vesicle goes and fuse with the plasma membrane and release that substance to outside. Okay. Then the plasma membrane regain its original form because that vacuole become the part of the plasma membrane, and at the same time that substance is released to outside of the cell. That is exocytosis. Exocytosis is the transport of material from cells inside to outside with the help of formation of vacuoles. Vacuoles, clear? Next, pinocytosis. Pinocytosis is a type of endocytosis. It is a bulk transport. Bulk transport of, this is important here. Bulk transport of fluid materials from outside to inside. The term means that I drink. And that pinocytosis is induced by appropriate concentration of the certain proteins, amino acids or certain ions in the medium surrounding the cell. If we consider that this is the cell, this is the cell, this is the cell. If the concentration of certain protein or certain ions are sufficient in the environment outside the cell, then the pinocytosis will happen. Pinocytosis will happen. It's a process, pinocytosis. If it is taking in the substance, how the membrane first form a pit, means it infold towards inner side, and then take that substance to inside that invagination. It then form a vesicle, pinches off from the plasma membrane. It pinches. Listen to that word. Pinches off from the plasma membrane and form a vesicle by entrapping that fluid matter. And that vesicle are of two types. Small size vesicles are there, and large size vesicles also there. If the vesicle formed is small size, then it is called a micropinocytosis. There the vesicle size is only zero point 0.1 micrometer. Then when it become macropinocytosis, when the size of the vesicle become 1 to 2 micrometer, that time we can call it as macropinocytosis. Next, I am going to explain you about the receptor mediated endocytosis, where it is actually becomes significant whenever the substance that need to transport to cells inside is present only in low concentration. In that time, the receptor mediated endocytosis function. Okay, that process we are going to see. This is the extracellular space. This is the plasma membrane. In plasma membrane, certain pits are present. They are called coated pit, because that coated pit is covered by certain molecules called clathrin. Clathrin. Clathrin means it is a type of peripheral protein. Clathrin I will show you later. And also along with that, certain receptors also present in the membrane. And the substance that need to transport to cells inside first come and bind these receptors. And after those molecules bind with the receptors, the receptor with the molecules move towards this pit. It is called coated pit because it is wrapped by other protein molecules, peripheral protein molecules called clathrin. Clathrin, I will give you the spelling. Clathrin. So that till that I am showing molecules binding, it is moving to that pit. This is the clathrin coat. And what next happen? The pit deepens, and it detaches from the plasma membrane to form a vesicle. It is coated with clathrin, and that vesicle is called coated vesicle. Coated vesicle. Then what next happened? That coated vesicle lose its clathrin coat. And that what happened to that clathrin coat? That clathrin coats released from that vesicle is recycled back to the plasma membrane. Okay. And similar other vesicles come together and fuse each other to form big vesicles called endosomes. Endosomes are formed. And from these endosomes, then that ligand molecules, we can call these molecules as ligands. Ligands molecules are detached, and it stay free in this endosome. And then again this endosomes moves much more deeper towards the cells inside, and it becomes widen and develops to invagination to divide the endosomes into two part. And in one part, what left? That ligand molecules. In another part, left that receptors, free receptors. And the part that is bearing the free receptors, it is called a tubular portion. Tubular portion. And that portion having that ligands is called vesicular. Vesicular portion. Then it detach separately, and this tubular portion it recycle back to the plasma membrane, and the vesicular portion proceed further to deeper portions of the cell. And what happened to this vesicular portion that's bearing the ligands? If it contains proteins or other macromolecules or other particles, it fuse with the, this vesicular portion fuse with the lysosome. Lysosome, and the lysosome enzymatically digests the ingested materials. It ingested all this ingested material. This is the process. I think now it's very clear to you. Okay. Next is phagocytosis. Phagocytosis means it is a type of endocytosis itself, but it is taking in larger quantity. That is the important thing. Larger quantities of the material than pinocytosis or receptor mediated endocytosis. It is described by E. Meshnikov, and here the vacuoles that is entrapping the materials that need to be transported, it is called a phagosome. It's a specific name for that vesicle. It's called phagosomes. The process is that the prey is first temporarily turned immobilized by the secretions from the phagosomes. Means from that vesicles it secretes some substance, and using that substance it paralyzes the prey temporarily, and then entrap the prey in that vesicle. They entrap. Suppose this is the prey, the vesicle, it entrap the prey in that vesicle and then digest it by keeping it inside the phagosome. This is the phagocytosis. Okay, with this we can finish today's class and section. Thank you.